Hello, welcome back to part two of the benchling demo. And today we're going to go through how to use benchling to simulate an assembly for the purpose of cloning, such as cloning an insert into a backbone, and as well as how to design and analyze primers in benchling as well. So using the example here, we have a plasmid that is a gene therapy plasmid. Actually, it's an adenovirus plasmid that we're going to replace the fragment of EGFP, which is a fluorescent protein, with a gene of our choice. So to simulate this process, we're going to put a M-cherry, which is a red fluorescent protein fragment, into the EGFP position. So to do that, first we will locate some enzymes around this gene that can help cut out the EGFP. So here we have HIN3 that sits nicely on one end of GFP. And when you look at the enzyme here, you can see that it shows you where it cuts, as well as when it's highlighted in purple, it shows you where its recognition site is. So HIN3 is a popular restriction enzyme that's used in cloning. And when we look upstream of EGFP, we can see there's a bunch of enzymes we can use, but we're interested in an enzyme that cuts specifically here and nowhere else in the plasmid. So EcoR1 is a potential option as well. So to check if they cut anywhere else, we're going to the find enzyme function to see EcoR1 and how many cut sites it has. This one only has one cut site, which is perfect for our application. And you can see that for other enzymes, some have like 21 sites, and that's not desired. Makes your life a whole lot harder when you have to deal with that. And then now we're going to check HIN3, which also has one cut site. Fantastic. That means we can move on and use these two enzymes to cut out GFP so that the plasmid has sticky ends on each side after the fragment has been cut out. And now the task is to design a compatible overhangs that can stick to this plasmid so that we can replace GFP with M-cherry. So let's go to the M-cherry fragment. I've already created a forward primer here. And so there's three components for us to think about. First is the part that actually anneals to your fragment. If we highlight this region, it's really nice that in benchling, it gives us a melting temperature estimation, the GC content, the length of the nucleotides that you have annealing to the template. In general, we want minimum of 18 nucleotides so that it has some specificity when it comes to binding. And if we open up this primer, this one I've already designed. So it contains a recognition site for the enzyme EcoR1. So when it recognizes this specific sequence, EcoR1 will come and cut right here. I also added three bases upstream of that restriction enzyme site as a good practice, just to make sure that the enzyme can come here and sit on a fragment. It's extra padding, essentially, for the enzyme to come to the recognition site. So that's our forward primer, and now I'll show you how we design our reverse primer as an example. So let's say we want to highlight 18 base pairs here. And here we can see that the GC content is 50%, great. Melting temperature, similar to the forward primer, so it's also acceptable. Then we're going to click Create, Primer, Reverse, and it's going to show us the bases that are actually annealed to the template. So upstream of this at the 5' end, so make sure that your reverse primer, you're not confused about where you're adding your sites. You want to add your HIND3 enzyme right over here. So you can look up the enzyme of your interest on benchling and it gives you the recognition site that you will use. So we're going to paste that upstream of here. You'll see that it gives you an error. That's because it doesn't like having the overhangs here without specifying how many overhangs you have because it doesn't stick to a template. If you click the up button, it will automatically recognize how many overhangs you have. So remember, we also want to have 6 base pairs upstream for the enzyme to sit on, just as we did in the forward primer. So we can just put in random 6 pairs, whatever could work, since it won't be translated as a part of the protein. And then we want to click the overhang button again to make sure we account for the actual 6 base pairs we added, so total of 12 overhang. And then we can now save the primer. So let's name it M Cherry Reverse. Save primer. And perfect. So you can view the primers you created by clicking open this arrow signature here that highlights it as primers. And you can see that both of your primers are here. So we actually have to link the primers together in order to simulate a PCR. As you can imagine, in this example, there's only 2 primers, but in a more complex experiment, you may have multiple primers. So in order to signify to the software which primers go together, we're going to highlight the 2 primers. We're going to click link primers to link them together. And great, so now we have the primers linked here. Now we can click Create PCR Product, which will gives us our PCR amplicon. So here we can see that, okay, now the primer has bound. It has created a cut site upstream of the M cherry for EcoR1 to recognize right here. And then also on the reverse primer, we have Hin3 recognition site also introduced. Great. So this PCR worked fantastic. The other thing that we want to check here is whether EcoR1 cut sites are found in this fragment. There's only one cut site, which is the site we introduced. Great. And Hin3, only one cut site. Perfect. So now we can move on to assembly. So to assemble the insert fragment into the backbone, which is our gene therapy vector, we're going to click Assembly Wizard and we're going to choose Digest and Ligate. So this is the simplest type of cloning using restriction enzymes to cut and ligate the insert into the backbone. So we're going to go back to the backbone tab. Now we're going to highlight everything from EcoR1 to Hin3. So we want to take the inverse of that. So let's do that again. EcoR1, which is somewhere over there. EcoR1 and Hin3. So to make this a little bit easier, you might want to show the enzymes that you're looking for. So EcoR1. Great. Perfect. So now we've highlighted all the region that we want to use as backbone. So then we can click backbone, set from selection, and it will show you what the cut site will look like, and it will give you an error if there's any problem. So now we're going to go to our insert, which is our PCR product. So we are going to do the same thing. We're going to click EcoR1 and Shift key, Hin3, to highlight the fragment that's actually going in to the assembly. We're going to click Insert, Step from Selection. And again, it will check if the sticky ends are actually compatible. Since we're doing this simple restriction enzyme cloning, this should work out great. So this works, and now we are going to click the checkbox, AAV and cherry, assembled. We're going to name our plasmid, and then it will create the plasmid for you. Great. And now we can see that instead of having a GFP here, we now have an cherry. So our assembly was successful. You could also see a gel simulation on here as well. So if we wanted to digest this plasmid to verify it before we send for sequencing, we can do run digest. And then there is a tab called Virtual Digest that allows you to see what it would look like if you cut it up into 2 fragments. So you have your insert and then you have your backbone. So with that, that completes the demo of how to create a plasmid and simulate an assembly in Benchling, how to create primers, link primers together, and simulate PCR, as well as a virtual digest gel. So yeah, looking forward to seeing you in the next one.